Hello everyone, warm greetings from my side. I hope everyone is doing well. And here I come up with the next installment of the DNA replication series. In my previous video, I have discussed about the various activities and various actions of the enzymes, playing different roles at different stages of the replication process in the prokaryotes. Where at the end, I have also added that in the upcoming video, that is in today's video, I will be wrapping up the entire replication process taking place in the prokaryotes. But after saying that, I realized that there is one single basic concept which everyone knows, but a few analyzes it. So before wrapping up the replication part taking place in the prokaryotes, I thought of explaining that very basic and simple concept. That is, the DNA replication process takes place in a bidirectional method, which does imply that on either side of the origin of replication, that is ORIC in case of prokaryotes, there is replication fork that moves ahead, and on either side of the replication fork, replication do take place simultaneously. So this is what we are going to discuss in today's lecture, that how on either side of the ORIC replication forks moves ahead, along with which the replication process also takes place spontaneously. So I hope you would enjoy this lecture. So let's begin our lecture without wasting any more further time. And so here we come to the bidirectional mode of DNA replication. I'm sure most of us know, as it is provided in written format in most of the literatures, that the DNA replication process is bidirectional. But the problem arises when the illustrations of the same basic concept in the form of diagram is given or is provided, where only half side of the story is showcased. Now in this diagram, which is provided in most of the literature, we can see there is a single replication fork, wherein the process of replication proceeds towards the movement of the replication fork, and at the end of the process we have two different double-stranded DNA, and in each double-stranded DNA we have one parental strand and one newly synthesized strand. But believe me guys, there is only half side of the story. So without any further delay, let's cover up the entire story and understand the same DNA replication process that is bidirectional in a much better way. So let's start. Now, as discussed previously, ORIC is the region from where the process of replication initiates, as it is this region from where the melting process do take place, and we know that melting is very essential for the process of replication to move forward. But the process of replication takes place in both directions simultaneously, which means, yes, we know that the process of replication, it starts or originates from the ORIC, but it originates or starts in both the direction from the ORIC. That is, on either side of the ORIC there is a formation of a replication fork that move in the opposite direction, and at each growing replication fork, one strand is known as the leading strand, and the leading strand is synthesized continuously from a single primer, and it is this leading strand which base pairs with the leading strand template, and this leading strand grows in 5' to 3' direction. The growth of the leading strand proceeds in the direction of the movement of the replication fork. The other newly synthesized strand, which is in the other side of the leading strand, is known as the lagging strand. The reason is very simple, because this newly synthesized strand, which is on the other side of the leading strand, is synthesized discontinuously and it requires multiple primers, but the leading strand requires a single primer for its synthesis. And on the other hand, on contrary to the leading strand, the lagging strand requires multiple primers, and that too, it is not synthesized continuously but discontinuously, and thereby it is known as the lagging strand. Now, having understood this, to understand it much more deeply, we need to go for a diagram. Now let's look at this schematic diagram. Now coming to this schematic diagram, this particular portion is a segment of the circular chromosomal DNA of the E. coli, which I have tried to draw, where in a particular region I have shaded red because I want to demarcate this particular region as the ORIC, or the region from where the melting begins. As the ORIC forms an open complex, it leads to the formation of two different replication fork on either side. Now, as you can see in the second diagram, there is a formation of two different replication fork on either side of the ORIC, one on this particular direction and the other on this particular direction of the ORIC. But in most of the literature, only half side of the story is revealed, where only a single replication fork is shown. But today we are going to understand the totality of the DNA replication story. So with the formation of the replication fork, the primase leads to the formation of primer, and as we know, it is the primer which produces or which provides the 3' OH end to the DNA polymerase, so that the DNA polymerase can elongate the DNA replication process. Alright. Now, as you can see that the parental strand, that is this particular strand, is from 3' to 5' direction. Therefore, the synthesis of the new strand would be from this direction to this direction, as this is the 5' end and this is the 3' end. So in the 5' end, there will be an addition of a primer. Now this primer will provide a 3' OH, thereafter which the DNA polymerase can carry forward the process. Now, on contrary to this particular site, moving to this particular site, as you can see that this side holds a strand which is from 5' to 3' direction. Therefore, the synthesis of the new strand would be from this direction to this direction, because this direction is the 5' direction and this is the 3' direction for the newly synthesized DNA. Now from the 5' to 3' direction, there is an association of primer, thereafter which the DNA polymerase has elongated the process of replication. Now the same story is going simultaneously at the other side as well. There is association of primer along with the activity of DNA polymerase on both the sides. The story is further cleared when we move on to the third stage. The third stage resembles the further opening of the replication fork. Now with the further opening of the replication fork, as we know that the direction of this particular newly synthesized DNA is from 5' to 3' direction. So therefore, a single primer, if provide the DNA polymerase with the 3' end, then it would be sufficient for the DNA polymerase to carry forward the replication process towards the direction of the opening of the replication fork. But coming to this side of replication, then a single primer is not sufficient, because at the initial stage only this much was opened. Now as this much was opened, from the 5' to 3' direction, a primer was provided. Now this primer provided DNA polymerase with a 3' OH, from where the DNA polymerase carried forward the DNA replication process up to this particular portion. But as the replication fork opened further to this particular part, now there was no RNA primer to initiate the process of replication. Therefore, again a primer was attached to this particular end, and which provided the 3' OH, because of which the DNA polymerase could proceed forward the replication process. And again, after some more time, the replication fork opened much more, and because of which there was a requirement of another primer from the 5' to 3' direction, so that the primer could provide the 3' OH, so that it could help the activity of the DNA polymerase to produce this newly synthesized DNA. Right? So here you can see that the newly synthesized DNA on this particular side has got only one primer. That is, one primer is sufficient for the synthesis of the new DNA strand. but here multiple primers were required. Now, the DNA strand or ddNA strand which required a single primer for its synthesis is known as the leading strand. And on the other side, the DNA strand which required more than or multiple primer for its synthesis is known as the lagging strand. It's very much simple. The side which required multiple primer for its synthesis is known as the lagging strand, and the side which requires only a single primer for its synthesis is known as the leading strand. So this much story we knew. Now moving to the other side of the replication form or to the other side of the ORIC, the story is completely different. The story is, now for example, on this particular side, the replication fork opened up to this much. Now since it's opened up to this much, now we know that on this particular side the replication process will begin from this direction to this direction, as this direction demarcated 5' direction, and this 3' direction as the parental strand is from 3' direction to 5' direction. So towards the 5' direction there would be a formation of a primer. This primer would provide a DNA polymerase with a 3' OH from which the DNA polymerase can initiate. But on the other side, for a parental strand it is 3' to 5' direction, but for a newly synthesized strand it is 5' to 3' direction here. So from the 5' to 3' direction there will be a formation of a primer, and the primer will proceed forward the activity of DNA polymerase, and the DNA polymerase will provide us with the newly synthesized strand. Now as the replication fork moved ahead, now in this direction there was a requirement of another primer so that the replication could start from 5' to 3' direction, so another primer was attached. So with the attachment of this another primer, the replication process carried forward. But here on the other side, a single primer was sufficient to carry forward the replication, as on this side it was the 5' to 3' end, and we know that with a single primer, is sufficient to carry forward the replication process from 5' to 3' end. Right? So what we have seen here, we have seen that on one side of the ORIC, if a particular strand is acting as a leading strand, then on the other side of the ORIC, that particular leading strand would act as a lagging strand. And on the other hand, on one side of the ORIC, if a particular strand is acting as a lagging strand, then on the other part it would act as a leading strand. So this is the bidirectional replication process. If you have loved this video, please do share and like at the same time. Thank you very much.